What's up everybody? So in this video we're going to be talking about neural signaling. There's quite a bit to cover, but to be honest it's not that complicated. It's pretty straightforward. I think for HL though, the HL only video, there will be a lot more detail and it will be a lot more complicated. But for the SL, HL, the core stuff, it's not that bad. So as usual guys, go check out teachme.org for a lot of really sick notes and lots of IB style questions, okay? Especially these questions, you guys need them to really nail the final exams, right? Get prepared for that. So without further ado, guys, let's just get started. So what the heck? Nervous system. So I've already told you so many times in other videos that our body, right, has so many systems, all of which have their own unique, different job, right? For example, we got our cardiovascular system, which is involved in our heartbeat, sending blood all across our body. We got our immune system helping us fight infections and disease. We got our digestive system helping us break down food and all that. Our respiratory system to help us breathe. So many systems. And here, guys, we've got another one, the nervous system. What the heck is the nervous system? You may think, nervous? Like, I feel nervous. Well, no, it's the system that's made up of nerves, which is a kind of cell. Okay, a kind of cell. Although your nervous system obviously is responsible for your emotions and that stuff as well. So technically it can make you feel nervous. But the reason why it's called nervous system is because it's a system made up of these cells called nerves or neurons. Now, what does your nervous system do? So there's a few very key things. Your nervous system, okay, as I just said, is made up of these little cells called nerves, okay, and they have some other names that you may hear. The key one is neuron. This is the name we use in science, neurons. It's the scientific name. You may commonly hear a teacher refer to it as nerve cells or nerve fiber, but the key one really is neurons. So your nervous system is made up of these cells called neurons. And these neurons, wow, look at them, man. They're looking crazy, man. They look mad, right? Like they look much different from a standard cell that you think of, but they're still a cell. They still got a nucleus, they still have organelles. They're just unique and specialized for their job. So what is their job? These cells, these neurons, they're special because they can carry little electrical impulses, or in other words, little electrical signals from one point in your body to another. They're essentially your control system. They are the messaging system of your body, telling what to do what and when to do it, and all of these kind of things. They control things, okay? That's what your nervous system is for. Okay, great. So we know our nervous system is the control center, but give me some examples, man. What exactly does it do? So I'll give you a few things which your nervous system is responsible for, okay? One big one is muscle control, okay? Yeah, you cannot fart without your nervous system, okay? Because your nervous system is going to send these signals to your muscles via these neurons and tell them to contract, okay? Also, when you get slapped in the face by some lady after you annoyed her, you're going to feel something, right? So sensation, your nervous system helps you feel things, okay? Interpret pain or pleasure, right? Sensation, that's very important. What else besides muscle movements and sensation? There's like a million more, but I'm going to mention this last one here for you guys. Emotion, memory, right? Without your brain and your nervous system, you can't remember things. You can't remember who that person is that you keep seeing. You can't remember, you can't have emotions. You can't be sad or angry. Okay, so there's so many things other than these, but these are some key ones I want you guys to be aware of, okay? Your nervous system. Now, as I said, your nervous system is just one of your systems in your body, but what exactly is it composed of? What comprises our nervous system? Okay, we can basically split apart our nervous system into two components, the central nervous system and then the peripheral nervous system, okay? Both of these combined forms your nervous system. So what exactly is our central nervous system, or your CNS? They abbreviate it very often as CNS and this one PNS, okay? It's too long to write out the whole thing the whole time. So what makes up your CNS? Two things, your brain and your spinal cord, okay? You can see here. And it kind of makes sense according to the name because these two are most centrally located, okay? In the midline of your body, right? And your brain. Now, what I want to say is, don't think of the nervous system. When you think of the nervous system, don't think only of the brain, right? Your brain is merely one component of your nervous system. It's merely one component of your central nervous system. So it's a very big part, but it's only one aspect. So don't confuse nervous system with brain, okay? So these are the two things making up your central nervous system. The brain, and attached to it is a spinal cord extending all the way down the midline of your body, protected by your vertebral bones and all that, so it's not that easily broken. And it has a purpose, and we're going to talk about the purpose just now. But for now, I want you to know that this is what's included in the central nervous system. Now, as you can see here, the peripheral nervous system, this is basically the components that come off of the central nervous system. Because you have your brain, right, and your spinal cord as the central nervous system. Now, all of these neurons and nerves coming off of your central nervous system, your spinal cord, and going to the rest of your body, like your teeniest, tiniest toe, or your teeny, tiniest finger, right, or your muscles for farting. All of these places, these nerves are considered a part of your peripheral nervous system, okay? So what do they include? Two big components: your motor neurons and your sensory neurons. So again, remember, neurons is just the name for the cell that makes up our central nervous system, right? Because all of these things, your brain matter, your spinal cord, these guys here, they're all made up of neurons, little nerve cells. Now, there are two kinds that comprise our peripheral nervous system. Two broad categories. It's a bit more complicated in reality, but for the IB, these are the two we need to limit you guys to: motor neurons and sensory. So motor, think of motor as like moving. It's doing something, it's activating, right? A motor is something that's emitting power, okay, work. So a motor neuron is a kind of nerve cell that travels from our central nervous system, and then that travels from our spinal cord and will go to our muscles, okay, to cause them to contract. That's why they're called motor neurons. Sensory would be the opposite. Sensory means sensation, feeling things. So these kind of neurons would go from whatever area of our skin or whatnot, and will travel towards our spinal cord, okay? So these are sensory neurons. Both of these combined, we can call peripheral nerves, okay? These two form our peripheral nerves because these are the nerves, part of our peripheral nervous system, okay? That's the basic sense. Now, what exactly is the purpose, again, of the peripheral nervous system? Then we'll go to the central because it'll make more sense that way. So the purpose of our peripheral nervous system, as far as you guys need to know, is two things. One, the function of the motor neurons, which I just said, is to carry information, an electrical signal, away from our spinal cord, our central nervous system, towards a specific muscle that needs to contract. On the contrary, our sensory neurons will carry information towards our spinal cord, towards our central nervous system, so that we can feel sensation. So what exactly is the function then of our central nervous system? Three key things. One, it can receive information. If your best friend pinches your arm, these sensory neurons are going to pick up that thing and send the signal towards your spinal cord, okay? And that's going to be sent towards your brain, okay? And then your brain, your central nervous system, is going to be able to interpret that information. Ow, I just been pinched, man, okay? And then your brain is going to think about that, and it's going to initiate a response. Are you going to slap your friend right back? Or what are you going to do? And if it decides for you to punch a friend, it's going to send a signal down the spinal cord, which will come off in the peripheral nervous system as a motor neuron to go and hit your friend, right? So that's how they work together. So you can think of the central nervous system as the big boss, okay? It's going to decide when a message is sent out so you can move your muscles, and it's going to decide how to feel about something. Because this guy, your peripheral nervous system, is kind of just like a worker for the big boss, okay? The big boss will decide, okay, let's move a muscle, and it'll send a signal, and then the peripheral nerve will go out and move the muscle. And on the contrary, if you feel pain, it will move the signal towards the brain, and the brain will decide how it feels about that and what it wants to do next. So the peripheral nervous system is really a worker of the big boss, okay? And this guy is the big boss. He's deciding what's what, and he's interpreting everything. So that's the very key things you guys need to know about the central nervous system and the peripheral nervous system. I want to give you guys a silly little example here, again, to wrap it up and make sure you guys understand the central nervous system and peripheral nervous system. I don't know how many of you guys actually like MMA, It's just a kind of fighting sport, you know. It doesn't matter. Here's the idea. Let's say this guy here, he's famous. His name is John Jones. He decides he wants to punch this guy, right? They're fighting. So his brain, or his central nervous system, will send a signal down from the brain into the spinal cord, down the spinal cord to a level, and then it's going to come out and extend. This is now the peripheral nervous system, right? This peripheral nerve will come out and go to whatever muscle he needs to contract to punch this guy. Now let's say he lands this elbow, okay? Now this guy's going to feel pain. There's going to be a nerve that's going to detect that pain, and this is the peripheral nervous system, right? And it's going to go towards the spinal cord and then send that signal to another nerve, which will take it up towards the brain. Okay, now he's going to feel that pain and be like, oh, what am I going to do next? Okay, he's going to interpret that information. And maybe he punches him back, maybe he runs away. Okay, that's the idea. That's what you need to understand about the central nervous system and the peripheral nervous system. Okay, hope that's clear. Okay, now, so we know now what the central nervous system, let's go back here. We know now what the central nervous system and the peripheral nervous system does, but we actually finally now got to look at the neuron structure. We know now neurons make up this nervous system, and it's responsible for all this stuff, sending all these signals. But what is the structure of a nerve cell? What's the structure of a neuron? So here we got it. Here's our little neuron. Remember, our neuron, right? This cell, this crazy looking cell, is an individual cell that carries little electrical impulses, or you can think of it like little electrical signals from one point in the body to another, right? Now, wow, it's crazy looking, right? I gotta admit. And there's a lot of little structures which you guys absolutely must remember, okay? Because on the test, they can ask you to draw the basic structure of a neuron or label a neuron. So it's very important, okay? Don't ignore this stuff. It's very, very key. So let's start off here at this top part here. It looks crazy, looks like someone who woke up with mad crazy hair, right? With one eye. Now, what is this guy here? What is this thing here? This is the nucleus, okay? We know every cell has that nucleus, right? Except like a red blood cell, but this cell has its nucleus right here. And what's this stuff surrounding the nucleus? Well, it's got some cytoplasm, right? And you can even see some little organelles, right? I didn't put a lot of organelles here, but there's a lot of other organelles ongoing here. I try to keep it simple. There are other organelles, but I'm only labeling the key things here to the neuron. So we got our nucleus, we got our cytoplasm, and some organelles. Now this whole area, okay, the combination of the nucleus and these organelles and the cytoplasm, we call that the cell body, okay? The cell body. So this whole area is the cell body. This is where most of the things are going to happen, where they make proteins and things like that, right? The cell body. Now, what are these little extensions that come off of the cell body, these little branches? We call them dendrites, and you'll see why they're very, very, very important later. But for now, you guys should know that basically dendrites are these little extensions that come off the cell body, and these areas are going to be receiving little electrical impulses or little electrical signals from another neuron. Okay, so one neuron will send the signal to this neuron here. It will transfer that signal here at the dendrites, and then the signal will come in through here and extend all the way down to the end of the neuron. Okay, now before we get to the end of the neuron, what else is going on here? What is this? You see the little blue part that's extending pretty much this whole long part. Okay, don't worry about these yellow things for now, but you see the blue like little long pipe. We call that an axon. Axon. That is just the whole little pipe through which the signal is going to be sent. Okay. Now, you notice? We'll come back to this just now. Okay. So you see the axon. It runs all the way down, and then we get these branches at the end. Very, very similar to these branches here, but they have a different name. Okay. Because this is like the head. We call this a cell body, and this is like the tail. Okay. But we give this a specific name. These branches we call, very simple, they're at the end of the neuron, so we call them terminal branches. The beginning of the neuron and then the end of the neuron, so we call them terminal branches. And then if you notice, these terminal branches have little blobs on them at the end. We call these terminal buttons, okay? Don't worry about these too much. You'll see later what's really going on here at the terminal buttons, but for now, just remember the name. Now, we skipped out on this area here in the middle. Remember, I said the axon is this blue thing extending through here, right? Now notice these yellow things. These are actually cells. Okay, these are little cells wrapping around the axon. We call these cells, okay, Schwann cells. Schwann cells, okay, Schwann cells. Look, they got a little nucleus there. These guys, they wrap around the axon. And when they wrap around the axon, they form many, many little layers. And we call that the myelin sheath. And you'll see later wow, that these myelin sheaths are really, really, really important, okay? For now, just remember that these cells here that wrap around the axons are called Schwann cells. And they, by wrapping around this axon many, many times, they form a myelin sheath. I'll show you a picture very, very shortly. Now, remember the axon. It's this long part and it's being wrapped around by these Schwann cells. Now, these little intermediate junctions separated by these Schwann cells. You see, there's a little junction here of axon, a little bit of axon there, a little bit of axon here. These are axon. These little pieces of axon, we give a name. We call it the node of Ranvier. Node of Ranvier. And again, don't worry about these things now, this little section here. We're going to talk a lot about how this works, what's really going on here. For now, we're really focusing on the structure. I want you guys to just look at the structure, look at the parts, and get familiar as much as you can. Now, okay, let's quickly run a little bit through here, okay? Run a little bit through here. So remember how I said, give me a split second. Remember how I said this axon, right? This blue little pipe, it's wrapped around by the cell. So in the very beginning, when this Schwann cell starts wrapping around the axon, it doesn't have many layers. As you can see, it's like one layer, okay? But over time, it forms, it keeps wrapping. It's like giving it a massive freaking hug, okay? And it gives it many layers. These layers we call myelin sheath, okay? That's very, very important. Now, for now, I'm going to mention one small sentence on it, but we're going to have a part on this later. But basically, the purpose of these myelin sheath, these Schwann cells wrapping themselves around these axons, is to speed up the conduction of this signal. When the signal arrives here at the dendrites and is being sent through to the other side, these guys help speed it up. It helps send that signal faster than the signal would send without these guys. Okay, so that's what you should know for now. They speed up transmission of this signal, okay, sending of this electrical signal. But we'll see how it works a bit later. Okay, now two last little bits here. How long do you guys think neurons are? Think about it. You know, if you go back here to this guy, if you have your spinal cord, and we know these motor neurons to flex your tiniest toe has to come out, you can imagine that a single neuron can be really, really long. Think about it. A neuron coming out of the spinal cord here has to travel all the way down to your smallest toe. So they can be like over a meter long. And then you can have other nerves that are really, really short, like a couple centimeters or millimeters, really, really short. So there's no standard size for a nerve, okay? So for a neuron, okay, they can really kind of go anywhere. They can be any kind of range, depending what kind of structure you're sending a signal to, how far that structure is. So there's a big range, okay? And the part that gets longer is this axon. So in some neurons, the axon will be pretty short, and in other ones, it'll be really long to extend all the way down to your tiny toe, okay? So that's very important. Remember, guys, as I already said this, I'm just going to put this here for you. Remember, the signal generally travels from the dendrites, this side, when it gets received from another neuron, and then gets sent through here, through the axon, all the way to the terminal button, and then from here, it'll be sent to the next neuron, okay? So that's just understand direction. It doesn't get sent in the reverse way. It normally gets sent from the dendrites down to the terminal branches, the terminal button, okay? So that's the structure, the anatomy of a neuron. Now I keep mentioning some words like nerve and neuron. What the heck's the difference between a nerve and a neuron? We know there's many names for a neuron, like nerve cell or neuron or nerve fiber. But what's a nerve? Like what exactly is a nerve? What's the difference between a nerve and a neuron? Basically, a neuron is a single cell. When you bundle together a lot of these neurons, you form a nerve. Okay, many individual neurons grouped or bundled together form a nerve. Let me show you here a nice little picture. So you see here, this is a scanning electron microscope. So remember, an electron microscope can look really, really close. And this one, the scanning one, can make it look very 3D. It's really, really cool. I like this kind of image. And you can see here, all of these little circles, that's an individual neuron. But look, they're all grouped together very, very nice and tightly. And this whole thing is called a nerve. Okay. So you can see the light blue part, that's the axon. Remember, going back to our anatomy, the light blue is the most inner side, that's our axon. And then the yellowy part wrapping around it, that's our myelin sheath. Okay, so let's label that clearly. So we got our, I missed it a bit, let me fix it for you lads. Nerve, and then the smaller individual neurons making up this nerve, okay? That's important. I hope that makes sense. And Here's another picture. This one is different. It's not a scanning electron microscope, it's a light microscope. So this one you can see, obviously they had to add dyes to make the colors appear like this, okay guys? It's not like naturally like this. So they have some dyes so we can see clearly. You can see all the little circles. What are those? Little nerve cells. But the group of them all together will form a nice big nerve. So a pretty good way for you two guys to remember this is, I don't know if you guys have this, but every so often I will bump my elbow on something and I'll have the shooting freaking pain, and we call that, we hit our funny bone, right? Well, we know in reality, what did we really hit? We didn't really hit a bone. We hit a nerve. There's a little nerve called your ulnar nerve, okay? And it runs right there by your elbow, and it's not really covered that well. It's not protected by bone or anything, so it's pretty obvious. So if you accidentally hit that region, it'll be a nice, sudden, unexpected stinging pain, okay? A shooting pain. So what did we hit here? A nerve. And this nerve is called the ulnar nerve. The ulnar nerve is made up of many, 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 many little neurons. So that nerve, the ulnar nerve, will have many, many neurons inside it, sending signals down to whatever area it's supposed to supply. Okay, so that's important to understand the difference between a nerve and a neuron. So guys, we've now covered everything up till here. We've covered now the structure, the big overview, CNS versus PNS. Now we gotta figure out how exactly this neuron actually works. How is this signal sent from one nerve to another? How is it transferred through the nerve? All of that little good stuff. We gotta learn this, okay? And that's like also another very big part of this video. But for higher level, you guys will learn far more detail. So if you guys don't completely understand this concept for standard level, it's partly because you guys should not be able to. We're not gonna give you nearly enough information to truly get it. But I'm gonna try and give you, tell you guys what you need to know and try and make it easy to remember, okay? But really, the amount you guys need to know for IB is very little for standard level, okay? So let's get into it. So how exactly? Okay, we got our brain here, right? We know our brain has a lot of these neurons, right? And they can send signals to other neurons and maybe eventually send signals to your muscles to contract, right? We know this is going on in our body. Now, how exactly is a nerve impulse or this electrical signal generated? How do we make this signal? Where does it come from? Let me give you a nice little story here. If you guys can remember this story, the real thing will be so much easier. Look here, what are we looking at? We're zooming into here a bit of the axon, right? That long little pipe. We're looking at the axon. So this right here is the inside of our axon, and this is the outside of our axon, okay? Now, we know it's just a cell. So what is this? This right here is the membrane of our cell, okay? The membrane, we know it's made up of like a phospholipid bilayer. Inside this membrane, we may actually have like a lot of these little protein pumps and these channels and all that, right? We know that's going on. Now, look at this little analogy. So imagine inside your cell, okay, inside your house. Let's say the inside of the axon is like your house. Inside your house is your family members, right? The people who belong inside these axons. Don't worry, later I'm going to tell you what these guys represent, but for now, just listen to this. So inside this axon, inside your house is all the people who belong in there, right? And then outside of your house, right, is all the people who don't belong in your house. They could be criminals and anything like that, right? Now, let's say this happens. These criminals, these people not supposed to be in your house, starts going into your house. They enter your house. What are you going to do? As these people start coming in, you're going to be in like a flight response. You're going to be like, oh, hell, I got to get out of here. I don't know if they've got guns or weapons or anything. So you end up leaving. Okay, so initially, these criminals, when they come in, right? And you notice people came in. So you start leaving. You're like, hell no, I'm going to get out of here. And you leave your house. Okay? And now guess what happens? Now you call the police and everything, and the police eventually come, and what they're gonna do is what? They're gonna get these criminals out of your house. Hopefully you can catch them and get them out of your house. And guess what? That's gonna allow you to be able to go back in your house, right? So this little analogy is very, very important, and you'll see if you can remember this, it'll help you a lot to understand the real mechanism. So let's now look at not the analogy version here anymore. We're going to look at the real thing. Okay, so here we got our same thing. We're looking at this little axon here. We got our inside and our outside of our neuron. We got these protein pumps and channels and all that. Let's label some stuff. So in this membrane, your phospholipid membrane of your neuron, right? You've got these channels and pumps. Let's name them. Let's name them a little bit. So this guy here, we're going to call that our sodium channel. This channel only allows sodiums to come through it, okay? Come through it. And then this guy here, Okay, it's going to be our potassium one. Potassium, K plus channel. So potassium can go through here. And then we got this last thing here. You guys learned about this in membrane transport. It's our sodium potassium pump. Remember, this was a pretty special thing. This guy was able to pump sodium and potassium in opposite directions, right? Using ATP. Okay, we're going to look at, so it's a kind of active transport. Both of these are like passive. So the molecules will go down their concentration gradient, all right? So that's what's in these membranes, a sodium channel, a potassium channel, and a sodium potassium pump. Now, very important part here. In a normal cell, in normal conditions, what happens is we have this sodium potassium pump. And so what it does is it pumps, what it does is here, this beautiful tip, Na, sodium, the cell does not want it, so we can say sodium, Na, Na, and we pump 3 of it. And because there's NaH, 3 things, we can put number 3 here. And then K is like, okay, I want you in the cell. And you can think of K plus as 2. K and a plus, 2. And Na plus is Na plus, as 3. So this will help you because the sodium potassium pump, what it does is it pumps sodiums out because we don't want it. Specifically, it pumps 3 out. So this guy is going to pump 3 sodiums out of the cell, out of the axon, and it's going to pump 2 potassiums into the axon. So guess what? If this guy keeps doing its job, pumping 3 sodiums out and 2 potassiums in, what are we getting? The outside of our axon will have a lot of sodiums and very little potassiums. And then the inside of our axon will have a lot of potassiums and very little sodiums. So you can see, because of this guy, normally in this is the scenario in our axons. We've got a lot of sodiums outside of the axon, outside of the cell, and a lot of potassiums inside the cell. Okay, that's very, very important. Pay attention to this. Now, if we had to measure the charge, because right, we're trying to generate an electrical signal. Let's measure the charge here. Because remember, this guy is pumping three positive charges out, because sodium is positively charged. So it's pumping 3 out and only 2 potassiums in. So you can imagine, after some time, the outside will be more positive compared to the inside, because we're pumping 3 sodiums out but only 2 potassiums in. So you can imagine the outside will be more positively charged compared to the inside, okay? Or we can think of it the reverse way. The inside will be more negatively charged, okay? Negatively charged compared to the outside, because there's more positive outside, less positive inside. So the inside is more negative compared to the outside. So remember, we're always comparing it to the outside. So the inside is more negative compared to the outside. This is the normal condition, okay? Wait 1 second here. Gonna get some words into your guys' brain. So you guys need to know a word called membrane potential. So membrane potential is the charge difference between across a membrane, between the inside and the outside. That's called membrane potential. The charge difference between the inside and the outside, okay? Now, we can call something a membrane potential is polarized if one side is more positive compared to the other side. Then the membrane potential is polarized, because you know polar means, like, if someone is bipolar, it means they have two extremes in personality or emotions, right? So polar just means extremes in something. So if we say the membrane potential is polarized, it means that one side, either the outside or the inside, is more positive compared to the other side, okay? It's polarized. It's not the same. It's not the same charge on both sides. That means it's polarized. Now, I know this, guys, that this is a bit much, but trust me, it's going to make sense here. Now, what is the resting potential? So the resting potential is this scenario here. It's the charge of the inside compared to the outside when there is no impulse, when there is no signal being sent, under normal conditions. Remember, under normal conditions, this pump is doing its job. It's doing this job all the time. So under normal conditions, this sodium potassium pump is establishing our resting potential. It's sending out 3 sodiums, bringing 2 potassiums in, and doing that consistently. And that is going to make the inside more negative compared to the outside. So that is our resting potential, okay? So when the neuron is not transmitting an impulse, the neuron is negatively charged inside. And this is thanks to our sodium potassium pump, okay? So this is our resting potential, like I said. Now, here we go. Now it's going to get a bit more nitty gritty. So this was the hardest part. If you understand this, now the rest is nice and easy. Okay, look. So now, remember how I said the one neuron will receive a signal from another neuron. So let's say this part here, this neuron here, these dendrites receive a signal, and we annotate that signal as a positive charge. So this positive charge is now arriving here. It's arriving, it came from another neuron, it's now transporting, and now it reaches this area here, okay, this positive charge. How are we going to move this positive charge down this whole thing? How are we going to move it down here? Because we want to send it all the way through the axon to the end, right? So how is this nerve impulse generated, and how is it moved across here? Look here. So what happens essentially is this positive charge is going to stimulate these channels, because guess what? These channels, these sodium channels, are very sensitive to charge. So as soon as a little positive charge arrives here, because remember, normally it's very negative in here, right? I just told you it's very, very negative in here. So if a positive charge arrives now, this positive charge is going to stimulate this sodium channel, which is normally closed because it's negative. But as soon as a positive charge arrives, now the sodium channel is going to open. And when it opens, what happens? Remember, there's a lot of sodium on the outside and very little on the inside. So if this door opens, naturally, these sodiums will flow right down their concentration gradient into the inside. And guess what that's going to do? That's going to make the inside positive. So this positive charge is now going to spread from here, where it activated the sodium channel, to here. Okay, because now all these sodium channels are coming in. All these positive charges are coming in. Okay? When sodium comes in like this, down its concentration gradient, we call that depolarizing. Because remember, the membrane potential was polarized. The inside was very negative compared to the outside. Now, all these positive charges are coming in to the negative side. So in a way, we're unpolarizing it, because we're sending in now positive charges to this side, which was negative. So we're trying to make it more even. We call that depolarizing. Now, when these sodiums are now in here, right, all of them come streaming in here, there's a positive charge, right, because of all these sodium, because of all these positive sodiums. Now, when it gets very, very positive, guess what? This guy now opens. So this guy opens when there's a little bit of a positive charge. Now, when all these sodiums comes in, it's even more positive. So you can imagine, right now, it's like very, very positive in here. Then these potassiums open, these potassium channel. And now, guess what? There's a lot of potassium on the inside. So it's going to very, very easily, what? Diffuse out, right? So remember, the sodium here was like our little criminals coming in. And now the potassiums is like the people living inside the house, leaving, right? They want to get out of there as quick as they can. And so they leave, right? So now, as they leave, guess what happened now? Now we have the opposite situation that we had when we started. Now all the potassiums are on the outside and all the sodiums are on the inside. That's the opposite scenario that we wanted. Because remember, all these sodiums are now in here and this positive charge hanging around here. Now, guess what? Before we move on, this positive charge, remember, all these sodiums come in here and they diffuse and they spread out. So this positive charge is now all over the place here. Guess what it's going to do now? This positive charge can diffuse a little bit. All these sodiums, as they diffuse and spread out a bit, they get close enough to this one here, this sodium channel. And now this sodium channel will open because it's sensitive to this positive charge. And when it opens, guess what? Now sodiums from here will come in. And now this area will become positive. And then the sodiums will spread again and cause the next part to happen again. So you can see that is how we spread this positive charge all the way from here to the end, okay? To the end of this neuron, okay? Very, very, very key. Now, where did we end? We ended here when we said that they're now at the opposite conditions that we started with. So now, here comes our police car, the sodium-potassium pump. It's going to now sort the situation out, remember? It's going to take these sodiums, specifically what? 3 of them, right? And it's going to pump them out. And then it's going to take 2 potassiums and it's going to pump them in. So if it keeps doing that, right, what's going to happen eventually? Then we're going to re-establish our what? Resting potential. We're going to reset our neuron that is now again negative. Let me just finish this for you guys. Send some more sodiums out, bring some potassiums in. So you can see now the sodium potassium pump has reset our thing to the original, where the inside is more negative compared to the outside. And now we are ready to receive another, let me put this here. Now we're ready to receive another signal. Otherwise, once the neuron, like if this didn't happen, then the neuron could receive one signal, send it to the end, and not be able to function again. But now we reset it so it can receive another signal and work again. Okay, now, so I just want to put this last one here for you guys. So we need to know the last word here, action potential. So all of these events here, this sequence of events that allows an impulse, this electrical signal, to travel through a neuron, that's called an action potential. So basically, to summarize it here, sodium ions coming in, potassium ions going out, and the sodium potassium pump resetting it, that's all part of the action potential. Okay, the sequence of events that allows an impulse, a positive charge, a signal to travel down. Okay, without these things, without this action potential, these events happening, we could not send this positive charge down the neuron all the way to the end. Okay, that's very, very key. So remember, a nerve impulse is the action potential propagated through the membrane. Okay, so this positive charge moving through the membrane, that's the nerve impulse. And it can only happen because of the action potential that's allowing it to. Okay, and last thing here, remember, I forgot to put the word here, when the potassiums go out, it's called repolarizing. Again, remember, I did put it here for you guys. Now remember, the sodium potassium pump, it's a kind of active transport. So this does require ATP, guys. Okay. So now we're done all the way up to here, right? We've finished now how an impulse is generated through this action potential mechanism and how that sends that positive charge, that signal, throughout the neuron from the dendrites all the way down to the terminal buttons, right? Now we got to figure out the next thing. How is this signal moved from the end of this neuron to the next one, right? Because neurons have to work together. So the signal will come here, it will travel through, and now it will like kind of lay it over to the next neuron. So that neuron can take that signal further along the pathway, right? So we got to figure out that. So we're going to talk now about synaptic transmission, okay? This one's also really interesting. I think it's much more easy than the concept I just explained. Like this whole concept is really, really tricky, especially at SL level, because you guys aren't given that much details and information and explanation. So a lot of it is really memorizing, okay? But at least for this next one, synaptic transmission, it'll be a bit easier in my opinion. So now that this signal, right, we explained how the signal was moved across with all this action potential mechanism, and eventually now it's moved to the end of the neuron. What do we call the end of the neuron again? Remember from the structure one, we call the terminal button. From here, it'll be moved to the next neuron. So we can see here, this is the terminal button. Sorry, the terminal button of one neuron. So I put it here. We call this the presynaptic neuron. And that will make sense very, very much now. Let me show you. And this one here is the next neuron. So we're zooming in really, really right there, okay? So what is this gonna be? That's gonna be the dendrite, okay? The dendrite of the postsynaptic neuron. Why does this make sense? You see this little space in between the presynaptic neuron and the postsynaptic neuron? Guess what this space is in between here? Based on the names here, postsynaptic and presynaptic. This must be the synapse, right? So this area here, right in between the two, the synaptic terminal button and then the dendrite, this area here is called the synapse. It also has another name called the synaptic cleft. Now this area is, as I just explained, the junction between the two neurons, okay? Or it can be the junction between a neuron and a muscle. Because remember, neurons activate muscles. So this scenario can also happen between a neuron and a muscle, okay? But in that case, when it's between a neuron terminal button and a muscle, we call this junction, it's still a synapse, but we give it a more specific name. We call it the neuromuscular junction, because neuro means nervous system and muscle refers to the muscle. So it's the junction between the nervous system and your muscle right there, okay? So now it makes sense why this one here, this neuron is called our presynaptic neuron, because it's before the synapse. Now this neuron, whichever one is trying to relay it to now, is going to be called the postsynaptic neuron, okay? Now, how does it work? So when this signal arrives, this impulse arrives to the end here, the synaptic terminal end, now it's going to do something. You notice here how on the terminal buttons, there are these calcium channels? Look here. they are also activated by charge. So when a positive charge, and in HL you guys learn about it, and it's going to be called voltage gated channels. These are sodium voltage gated channels, potassium voltage gated channels, and these are calcium voltage gated channels because they're activated by charge. Now, when this charge arrives here, it's going to activate these channels to open. Now, guess what? There's a lot of calcium on the outside, but very little calcium on the inside. So they're going to diffuse in. Now, when they come in, when they diffuse in, oh yeah, I want to put the steps here. So step 1 here is the impulse arrives. I have a word summary for this, so don't worry. So step 1 here of synaptic transmission is the impulse arrives at the end, the synaptic terminal end. Step 2 is sodium channels are opened, and calcium channels are activated by the charge and they flow in. Now the third step is these calciums, what they do is they have a way of transporting these vesicles. So these little bubbles here that contain little molecules in here, these are called vesicles, and they contain a specific molecule called a neurotransmitter. And I'm going to label it here for you guys, but we're going to talk more about what exactly neurotransmitters are. Okay, so they contain these little neurotransmitters. Now calcium is responsible for guiding these vesicles which contain neurotransmitters to the end here, the presynaptic membrane, okay? Right here, the membrane here called the presynaptic membrane. So it's going to guide them towards there. Okay, now, after it guides them towards there, now these vesicles, right, which contain these neurotransmitters, will fuse with the membrane and release the contents through a process called exocytosis, right? You guys learned about that in membrane transport. Now it brings us now to the point of what exactly are these neurotransmitters? These are little chemicals, okay, that allows us to send this positive charge to the other side. And you'll see exactly how that works. Because at the end of the day, we want to somehow move this positive charge through one way or another from here to here, okay? But how do we do that? We need these neurotransmitters, okay? These chemicals will allow us to send these signals between the two nerves, okay? But how? How does it do it? So after these, let me put the stage there. It's stage 4. Okay, so now these neurotransmitters are released and they're going to diffuse, right? Because right here, they're in a very high concentration. They're all compact. Now they can diffuse across the synapse towards the other side, right? Spread out. This is natural, okay? Now they spread out and go towards the other side. Now, I want to emphasize this. There are very, very many different kinds of neurotransmitters. Many different kinds in our body. There's only one that you guys have to know about, so I'm going to label it here, okay? And this neurotransmitter is called acetylcholine. This is one specific kind of neurotransmitter that IB wants you to know about. But no, we have so many, okay? And they have a very important role in sending this signal to the opposite side. If you have problems with your neurotransmitters, like the levels are wrong or you're missing one, then you can get various diseases such as schizophrenia, depression, many kinds of brain disorders. So these neurotransmitters are very important in keeping our nervous system intact, okay? So just know, you need to know this one kind of neurotransmitter called acetylcholine. Now, this neurotransmitter is released and it's going to basically diffuse across to the other side, the postsynaptic membrane, where it's going to bind to a little receptor. So a receptor is like a little binding site, okay? That's all it really is. It's a little binding site. Now, once it binds to this binding site, let me move it around. Upon binding of acetylcholine to this receptor, okay, it causes this channel to open. Okay, so that's different. Remember these channels, they were opened by charge. Remember, they're opened by this charge. This channel is stimulated by a chemical, okay, called a neurotransmitter. So these are chemically gated ion channels. When a chemical binds, they open. So when it binds, now this channel is open. Now remember, outside of the cell, what do we have? A lot of what? Potassium or sodium? A lot of sodium, right? We got a lot of this sodium floating around, right? So now if this channel is open, if there's a lot of sodium on the outside and very little on the inside, what's going to happen? The sodiums are going to go in. Does this remind you of something? Something over here. When the sodiums diffuse in, what happens? Now the inside is very positive. See, magic. Just like that, we indirectly sent this positive charge from this side to this side, okay? Okay, That's very important. So we really depend on these neurotransmitters. Otherwise, we can't send a signal between neurons. Okay, we're almost there. That was step 5, by the way. Binding here and causing sodium to diffuse down its concentration gradient. Now, we're not done though. You see these neurotransmitters. If we keep them here, that's a problem. Because if they keep activating this neuron, guess what? You're going to go into a sort of spasm, okay? Because your neurons are continuously firing nonstop. You're not stopping to activate these receptors, so these ion channels are always open, causing consistent firing, and you can get like cramps and a lot of mental disorders from this. So what happens is there's a little enzyme in this synapse here, which is responsible for grabbing these guys. So after they're released, they can bind, and after they've bounded and done their action, this little enzyme here, okay, specific for this neurotransmitter, is called acetylcholine esterase. Okay, because this enzyme, ACE, as you know, all the ones that end in ACE is an enzyme. They will bind this neurotransmitter called acetylcholine and break it down. Okay, now this broken down form can be taken up into the presynaptic neuron and be recycled and reassembled into a neurotransmitter so that the process can be repeated. Okay, so it's important to know that there are enzymes that can recycle, break down these little neurotransmitters into their components and allow them to be reuptaked by the presynaptic neuron and reassembled into proper neurotransmitters so the process can repeat, okay? Otherwise, there'd be consistent firing and would never end, and it'd be a problem, a serious problem. Give me 1 second. Cool, so in a way, there's six steps, very key. And I'm going to show you now here. I hope that makes sense, okay? So that's how we transmit a signal between two neurons, okay? So here's the word summary of that. I put 6 steps here as I labeled them. So here's all the words you guys need to know. So make sure you know all these things, especially the keywords here. And it was just by coincidence. Look here. The shape here looks like a neuron. It happened as we were making this, we noticed it just happens to look like a neuron, which is a neuron cell body, which is pretty cool. So the next important thing we need to talk about is speed of impulses. So we know now the structure of a neuron, we know how this signal is formed and how it's transmitted all along a neuron and how it's moved from one neuron to the next. Now we need to know which things can affect the speed of this process. What makes it slow, what makes it fast. So look here, again, we got our axon, we got the outside on the inside, but now we're making it a bit more realistic. Remember I told you that neurons can have this myelin on it, right? Which was a Schwann cell, remember? Let me label it here, myelin, right? It was the Schwann cell wrapping itself around the axon. And it had a very important job, and I told you it was to speed up this process. Now, how does it do so? Let me show you. So you know the membrane, right, has all of these little channels and stuff like that. And by the way, while we're at it now, remember that this is a 2D diagram. In reality, it's more like a pipe. So you can imagine all of these channels and pumps are located in a circle around the pipe. So it's not just at the top or at the bottom, it's like in a 3D kind of formation, okay? So hopefully you understand that perspective. It's not that important to worry about though. Now, how does this speed up this process? So in this area where myelin is, there is no channels, okay? No channels. So there's no movement of things in or out at these zones here, okay? So what that means is when the positive charge gets achieved through the action potential mechanism that we talked about earlier, it cannot activate any channels here because there's no channels here. So when when this positive charge spreads, okay, it has no choice, okay, as it spreads, but to activate the next area here where there is sodium channels. Okay, so let me put it here for you guys. Sodium, potassium channels and all that. Okay, it has no choice but to skip this area and activate the next one. So you can see the myelin, it acts as a sort of insulator to this charge. This charge, when it arrives here, it goes inside. There's no movement in or out. It insulates the charge. It keeps it inside the axon and forces it to go all the way to this side and activate the next segment, okay? And now here, the sodiums can come in again, the potassiums can come out, right? The action potential can repeat. So remember, these little zones in between the Schwann cells, or these myelins, are called nodes of Ranvier. Nodes of Ranvier, remember that. The little short bits of axon, right? In between the myelin is called nodes of Ranvier. So you can see here that through this mechanism, essentially what's happening is the charge is bouncing, okay, from one node of Ranvier to the next one. It's kind of skipping this area here, so nothing is happening in the areas where there's myelin. So you can see that this saves ATP and time. Why? Because if there's no pumps and no thing like that here, there's no sodium potassium pump. Remember, the sodium potassium pump uses ATP to reset the resting membrane potential. So there's no pumps going on here. We're saving ATP, but we're also saving time because now the charge can bounce and skip these areas. Normally things would have had to happen here, but now that there's myelin, we pretty much managed to skip these areas, okay, and conduct much faster. So myelin allows us to increase conduction and saves us ATP and time, okay? This kind of conduction, I crossed it out here because I think I'm 99% sure this is only for higher level. So I'm putting the word here in case you guys see it, but I'm 100% sure, 99% sure this is only a high level thing. But this is the name given to this idea of bouncing between nodes of Ranvier is called saltatory conduction, okay? So that's how we managed to speed up this process using our myelin, okay? That's pretty cool. So you guys need to know this. So this was one way in which we can speed up nerve impulses, right? We can, myelin can conduct action potentials faster than non-myelinated axons. So when myelin is present, the signal will be sent faster. In areas where, in neurons where there's no myelin, the signals will be sent slower. So that's one very important way. Another one, which you guys need to know, is the diameter. So you see the diameter of the axon. The larger the diameter, meaning the more space here is, the faster the signal is sent. Okay, you guys need to remember these two things. There's no need for an explanation. You guys don't need to know the mechanism behind it. It's very, very complex and it's beyond the scope of biology students. So just know that both of these things can speed up nerve impulses. A greater diameter of the axon and the presence of myelin. So we covered everything that you guys need to know about the nervous system. There was a lot, but I hope it makes as much sense as possible. Now that is really it though, but there is one component, one thing that was introduced in this chapter as well, which we need to cover because I don't know where else you guys will learn it, if your teacher will teach you it in some separate class or something, but it's a little thing that we need to learn about case-based questions, right? It's gonna help you to interpret graphs and things like that and help you put the right answer to get the points, okay? So let's take a look at it. We'll spend a couple minutes on it. It will really help you. Because a lot of people think they understand this and then they go to the test and they mess up and they don't know why. Okay, so let's look at this. It's the topic of correlation. Let me give you an example. Let's say this. My theory is that the more you study, okay, the more hours you study per week, the higher your grades will be. So in this example, the independent variable, the factor being changed was what? The amount of hours that you study. You can study 1 hour, 2 hours, 3 hours, nothing, whatever, right? And the thing we're measuring is your score on the test. Okay, so let's take a look here at the graph. Okay, here's the graph. It's not very realistic, but just bear in mind. So we got x-axis here, y-axis. So normally the x-axis is the thing we're changing. So for example, the more we go this way, that's the more hours we're studying. So over here, it'd be like 0 hours. And over here, 1, 2, so on. Okay, so the x-axis is representing the independent variable, the thing you're changing. Now the y-axis is the thing we're measuring, so the test scores. So you can see here, as we go this way, the more we're studying, okay, what's happening? The higher the grades are going. The more we're studying, the higher the grade is. If we study this much, your grade will be here. Study this much, your grade will be here. Okay, so the x-axis represents the independent variable, and the y-axis remembers your dependent variable. Why am I talking about this? So here's the example that I just mentioned. Because you guys, when they show you a curve like this, one thing they may ask you is, describe the graph that you are seeing. And then you shouldn't say things like, there's a line going up. If you say that, you're right. There's a line going up at a slant, but you're not going to get the point. The IB wants you to use the right words. So if you see a graph like this, where there is a very, very good correlation, you can clearly see it's going up like this. So the more you study, the better your grade will be. It's very, very clear. The data points clearly show it. In that case, you don't say it's going up. You say there is a strong positive correlation because positive means going up. So this is the word indicating that it's going up. So a positive correlation implies that as you increase your independent variable, so does the dependent variable. That's what positive means, okay? As you increase the independent variable, so does the dependent variable. So it's going to go up. And the word strong implies that these data points are very, very representative of the line. It means they're very, very close to the line. So it's very, very reliable, okay? So this, when you see a graph like this, you should say it's a strong positive correlation, okay? Now let's look at another one. How does this one look? It looks very similar to this. We know it's positive. It's going up, right? But it's weak because the data points are not that consistent to the line. They're not sticking that close to the line. It's more like I can kind of see there's a line, but it's not that strong. So in that case, you say it's a weak positive correlation, okay? Very important words to use. These will get you the points. So you can see a good example is hours of sleep and height. So they're saying here, the more hours you sleep, the taller you'll be. This is, you can see the graph shows a weak positive correlation. And that makes sense because hours of sleep is not the only thing determining your height. There are other things like your diet or your genetics, right? So therefore there's a weak relationship, okay? It's not a very strong relationship. Let's look at one more. Can you see any trend? No, so this is called no correlation. So don't say nothing, say no correlation. And an example would be shoe size and intelligence, okay? Because people with big feet can be stupid or smart. People with small feet can be stupid or smart, okay? So there's no correlation for this one. That's very, very key. So now the key thing here I need to address here, it is very subjective. So if I give you a graph that looks somewhere in between this one and that one, what will you say? Will you say strong positive or weak positive? You won't know, right? Because that's very subjective. It's up to the person. Therefore, they invented this way to quantify it. So they made a thing where they call it R. They denote it by R. So the correlation coefficient, or R, will denote the strength. So they'll basically give you the strength in terms of a number instead of just looking at it and guessing. Okay, so the number is between 0 and 1. The closer the number is to 1, that means the stronger the correlation is. So this one here would be 0, and this one would be close to 1. Not perfect, but very close to 1. Okay, that's very, very important to know. Now, if you square this R, so whatever value you get for R and you square it, that will give you the variance. It will tell you how closely packed these dots are to the line of best fit. So you can see this one's variance, or R squared, will be very, very close to 1, meaning they're very, very close to the line. This one's R squared will be very, very low because the dots are very far from the line. Okay, so those are two key things you need to know, R and R squared. Okay, let's look at one more thing. So use these words when you do the case-based questions. Don't say there's a slant. You're right, but you're not gonna get the point. So let's look here at the next one. So what's this one? It's going down. So now we say negative correlation. So this is a strong negative correlation, okay? It's not sloping down, it's a negative correlation because as you increase the independent variable, the dependent variable decreases. So it's a downward sloping. Next one. This one is the same thing but weak, okay? Because the dots are not so close to the line of best fit. And again here, no correlation. The difference here is when we look at R, instead of being 0 to 1, now it's 0 to -1. Okay, so when someone tells you the R value is -1 or anything negative, then you know it's sloping down. If the R value is anything positive, then you know it's sloping up. Okay, so it's either between 0 and 1 if it's positive or 0 and -1 if it's negative. Okay, so that's what you guys need to know. So that's it for this idea. Make sure you use these words. They can be really helpful to not losing stupid points. So now let's do some 3 questions. Okay, where do sensory neurons send signals to and from? Try it yourself, guys, and the answer is A. It sends it from your central nervous system, to your central nervous system from the sensory receptors, the areas that pick up the pain, okay, in your peripheral nervous system. Now, which neuron structure receives an electrical impulse from another neuron, right? One neuron will send the electrical signal from its terminal bulb to the dendrites. So it's going to send it to this one's cell body, and this one will send it down here, and then from here it will be sent to the next one's dendrites. So it's going to be A. Last one here, by means of which mode of transport are neurotransmitters released from the presynaptic neuron into the synaptic cleft? Do you remember the vesicles, they fuse with the membrane and release it by exocytosis. Then those neurotransmitters diffuse across to the other side, to the postsynaptic membrane. Okay, so I really hope that was useful, guys. There's a lot, and I really hope it helped you in some sort of way, and I will see you guys in the next one.